It's my pleasure to present today's first speaker, who is Diego Vidaura, who, after studies in computer engineering and then a PhD in machine learning and statistics in Madrid, decided to put all his knowledge to the service of neuroscience and moved to Oxford in a computational neuroscience lab. And since a few years, thanks to ERC starting grant, he started his group in Aarhus in Denmark, which is a beautiful place, by the way, and where they apply machine learning and statistical tools to problems in theoretical and clinical neuroscience. And today we'll talk about hidden Markov model and beyond, which makes sense because being hidden, there is some beyond.
So I couldn't come in the morning. There was some talk about HMM yesterday. So obviously I don't know what was. It's like this. I don't know what was said there. I assume they went through the basics nicely, but nevertheless, because I understand the nature of this conference is such that people can jump from one thing to another, it's not necessarily the case that everyone that is here seeing this today was also yesterday. For that reason, I think and because I have an hour anyway, I will start with basics, really slow and easy. And then this beyond bit, I will talk a little bit about things that are a bit more newer or advanced, if you like, for everyone's entertainment. All right, I'm going to start with an example that I use sometimes. Yeah, let's assume we have this guy who's I don't know anything about baseball, but I think he's very good, one of the best ball throwers in the NBA or whatever. And he's, we ask him, we have this thought experiment where we ask him to throw the ball and then to throw it again and then to throw it again. And the thought experiment is we ask him to try to throw the ball exactly in the same way every time. Just have the trajectory of the ball as similar as possible between the throws. And because he's very good, the trajectory is going to be relatively similar between trials, but it will never be the same. And why is that the case? You can appeal to weather conditions, wind, that he gets tired also. But the real most fundamental reason, because that's the case, is because the brain never does the same twice. So even if there's no wind, even if the guy doesn't get tired, the ball is never going to repeat the same trajectory twice. And that is not just randomness on environmental reasons, it's because the brain is really doing something different every time. So if you look at traditional methods to look at brain activity, so that variability, which I think is critical to understand cognition anyway, is lost because all these methods are based on across trial averages. So the classic, very classic, is the ERP. So you get signals, and then you get the average, and you look at how these average aspects of the signal behave. But anything that departs from the average is ignored as noise. If you want to be a little bit more sophisticated, you can use decoding analysis, which is basically a multivariate predictive version of the ERP. But again, it's the same, right? So you just stack across trials, and you predict using information that is, by definition, the same across all the repetitions, ignoring everything else. So the goals of the hidden Markov model, there's many goals about the hidden Markov model that you can think of, but for the sake of this talk and for the sake of this field, the goals of the HMM will be precisely to characterize time point by time point, brain activity, and trial by trial aspects of the signal, and how those relate to behavior. A second thing that I will reserve some time to talk at the end is that we can also use that to relate to subject traits in ways that are principled and handy, but I will talk about that later. I'm putting these two pictures of an MEG or an EEG and an fMRI because the HMM is meant to be sort of general, so we use it across modalities, as you will see, but I will focus on electrophysiology, not necessarily EEG, but MEG EEG for the sake of analysis in this case, they're quite equivalent. So ideally, what we want is to, I'm talking about this, variability and the information that we want to characterize. Ideally, what we want is to know the state of the brain at every particular time point. By state, I mean everything, every statistic or every characteristic that you're interested in. So, for example, in the case of EEG, it could be oscillations, or could be phase locking, or could be something that you think is interesting neurobiologically. In the case of fMRI, it could be functional connectivity. But nevertheless, whatever it is, you want to have a description of what happens at that time point. Of course, that's impossible. You cannot have a description of anything with just one time point. So the classic way to go about this is to use sliding windows. So instead of just looking at one time point, you look at a window around that time point where you can use those different time points to estimate things. And then the way the sliding window works, as you all know, of course, is that you choose the size of the window, you compute whatever you're interested in, then you move the window, compute it again, and so on and so forth. So you have a moving picture of whatever statistic you're interested in. The idea of the HMM is instead of doing that, which by definition looks at a subset of data at every time point, every estimation, is to look at the whole data at once, and then assuming that you can describe the time series using visits to a discrete number of states, then you describe this the time series throughout this finite number of states, which every one of them is characterized by a certain probability distribution. So you have the capacity of looking at the whole data at once, which gives you statistical power and so on and so forth, and you are sensitive to things that happen quickly because state changes can be quick. So that gives you a bit of an edge over a sliding window in that sense. So I'm going to very briefly describe the HMM if you don't know it, and then I will talk about how you can use that to describe trial-by-trial variability or time-point-by-time-point variability. So then the aspects, different elements of the HMM are three. So one is the state time courses, so when the state occur. These are probabilities. The next one is the straight probability distribution. So the HMM is in itself a probability distribution. It's a probabilistic model. It's a Bayesian model. So every state is also a probabilistic distribution. So the HMM is a nested hierarchical probabilistic model. And what distribution that is, you can choose depending on the type of data, the kind of characteristics you're going to capture. For example, in EEG, you could choose something that captures spectral properties of phase locking, whereas in fMRI, you might choose some probability distribution, like a Gaussian distribution, that captures functional connectivity, for example. And then you have the third element, which is the transition probability matrix, because the HMM is a dynamical system, very simple one, but a dynamical system. You also have the probability of transition between one step to another. So all of these three elements get estimated together using some process of Bayesian inference. Specifically, we use variational inference. And all of these get estimated at once. So as I say, you can choose different state distributions to adapt to different modalities. And that's a kind of a useful way to link across modalities, actually. So that's an example paper where they use an HMM to link fMRI and EEG using different observation models or probability distributions for the states. So that's useful. So that talk is titled how it's the word behavior in it. So is specifically focused on task data and on how to associate HMM descriptions to behavioral data. But the most common and easiest example of how to use the HMM is on continuous data. So I'm going to start just for illustration with an example from continuous data. So you don't have a task. So you have present state and you have the whole data set, the whole time series, the whole session. And then you run the HMM on that. So I'm just going to show an example. This talk, by the way, is not based on results. So I'm only showing results as examples. But the purpose of this talk is more like, it's more of a methods talk. It's meant to be useful. So I give this example just to illustrate. So we have this study where we had resting state in MEG, and we were interested in the default mode network. So we devised a type of HMM, so probability distribution for its states, that was able to capture power across different frequencies and phase locking, again, across different frequencies. So we ran that in the data. We found a number of states. So each of these states is, as I say, characterized by power and phase coupling. And that is given as a function of frequency. So here I'm showing 3 frequency modes, which are computed in a sort of data-driven way. So they're not canonical alpha or canonical beta, but they resemble as such. And we call them, just because of the areas they were involved in these states, we call them anterior higher order cognitive and posterior higher order cognitive with different frequency characteristics. So one, the anterior is obviously anterior regions and temporal poles, mostly in slow frequencies. And the other, the posterior higher order, is more like angular gyrus and parts are more posterior, obviously, and primarily on alpha. So the critical insight from that study is that the default mode network that we see in fMRI all the time, we don't see that in MEG that clearly. And in fact, when looking at these high time scales, it actually fragments into 2 different networks. And we saw that if you overlap these 2 networks, then that has a high correlation with the fMRI default mode network. So that was a suggestion that maybe the default mode network is not just one single network, but actually 2. But you need to look at higher temporal scales to be able to see that. So that was an example of use of the HMM on continuous data. But as I said, the point of this talk is task data. So we have, assuming you have an experiment, you have a condition, you have some design, stimulus being presented or whatever. And then how can we relate these HMM descriptions to task data? How can we leverage these things to learn something that we couldn't learn using a standard analysis like the coding analysis or the ERP using the HMM? So there's basically 3 ways to go about this in increasing complexity. So the first one is to run the HMM in an unsupervised manner. By that, I mean not including information of the task at all. So just run it as if it was continuous data. And then post hoc, look at how those state transitions and state probabilities relate to the task. So you can do sort of, we call them state evoked response analysis. So instead of having an ERP, you have an average of the state time courses. So the difference with the ERP here is that because the HMM states are sensitive to higher order statistics, like for example, connectivity or phase locking across different frequencies, you could have like an average description of that, doing that trial average. But that obviously, as you can imagine, has, well, it doesn't solve the question of between trial variability at all, because you're taking an average at the end. And the second thing is that you might be missing things about the task if those are a bit subtle. So the HMM is, when it's unsupervised, it tries to describe variance. So it tries to look at the things that are more salient in the data, but that those are not necessarily the ones that contain the features that you're interested in. So that you might be missing things using this approach. On the other hand, it's very simple. And it's, I think the things is most used by people. This is an example by Andrew Quinn in Oxford. He just has a tutorial paper on how to do this. So basically it has a task, then runs the HMM and finds this state about responses as if it was an ERP. So that's straightforward and fine. For the second and third, which is run it in a unsupervised manner, but with explicit associations to the task data, I'm going to talk briefly about the coding analysis. So I guess you all know this, but just in case. So the coding analysis is basically, so you have brain data, you have a condition, whether the subject was seeing a house or a face, and then you try to predict the task information using the brain data, using some machine learning algorithm. So that's the code analysis, which is based on this assumption of synchronicity. So you stack all the trials, and then you look at a window or a time point and you do your decoding analysis, cross-validate it, then you move to the next time point, you move to the next time point, and when you get an accurate prediction, then you reason the brain must be doing something at this time point. It's straightforward, very common, very used. Okay, so how can we combine the HMM and the coding analysis to get something useful? So this study that was not published, we're looking at mice, data from the hippocampus, where they're doing quite a complex task. I'm not going to get into the task. So we have LFP in the hippocampus, and they have spikes. So what we did, and that's the approach I'm talking about now, is just you run the HMM as unsupervised, so you don't include information of the task, as in the previous case, and you get state visits and state descriptions. So here we did that, found two states with two different descriptions of theta. So that task is not a navigation task, but is a task where the rats have to make some cognitive maps of the environment and reason about that. So there's a strong involvement of theta of that. So we ran this and we found moments where theta was much higher and a bit higher in frequency, and we found moments where theta was lower and a bit lower in frequency. So these two states, with relatively fast transitions between them. So that's fine, traditional way of running the HMM. And then what we did is to train decoders on each of these states separately. So per time point, the two states divide the trials into two groups. So we ran the cross-validated decoding on each group separately. So we are using the HMM to find between trial variability and relations to behavior in two steps. So the critical insight from, I'm not going to go into the neuroscience insights of that, but because we're still thinking about that actually. But the critical result here is that when you do that, the accuracy goes much higher for both states, even though they have fewer trials. So you have less data to train on. Nevertheless, in a cross-validated way, that's very important, you get higher accuracy than if you take the whole set of trials. So what does it mean? It means that at least there are two modes in which the rat is working, and this decoding works very differently for these two modes. So the way neurons encode this stimulus changes depending on which state of theta you are. So that's an example of a not very complicated way to look at between trial variability using HMM. Of course.
So you're in the red state for like a second, and then in the yellow state for half a second. What do you do with time in this case for the decoder? You collapse over time?
You do per time point.
But you have different amount of time points per state visit.
You have, so time point 100 milliseconds. You got, like, for example, here, you see that this and this, so the yellow and the red is the amount of time is spent in each state. So you go 100 milliseconds, and you have 40% of the time is one state, 60% of the time is the other state. You do the decoding on one, you forget about the other, and then you do the decoding on the other, you forget about the one. It's exactly standard decoding, only that you split the trials in two groups. And you do that time point by time point. You don't do the decoding across all time points. So you do as you normally do in decoding, time point by time point.
A given time point, you say I only consider whenever in this time point I was in state A.
Correct. And then you have two sets of betas or regression coefficients, decoding coefficients for each state per time point. And you see those betas are very different. And they're not very different because one is noisier than the other, because that would be an almost trivial explanation. Oh, the rat is distracted, didn't do the task well. No, actually, both decoders are better than the average or the global decoder. So that is a relevant result. Right. So the third way of including task information is to actually include it in the HMM. So instead of having two steps, you just have one step where the task information is included in the HMM. So in this case, every state is a decoder itself. So instead of having one decoder per time point, you have, imagine that there are 8 states, 8 decoders. So you trade having fewer decoders for letting them adapt temporally per trial. So you're fully leveraging between trial variability. So you can imagine, that's a cartoon, it's not real data, but you can imagine if you care about the visual system, and then you have a stimulus being presented, and there's a cascade of visual processing going from V1, well, I mean, thalamus, whatever, to V1, and then on the hierarchy. You can think of being that the point of this is to describe this progression throughout the hierarchy in a way that is per trial. And you can find differences in the speed by which you traverse throughout this hierarchy at the trial level. And that temporal information, those temporal differences between trials, is what we are interested here. So that's a cartoon. I'm going to show a real example. So we have this task where the people is shown a certain angle, and they have to remember they were shown an angle in the first place, and then they're shown angles while they are scanned, and they have to press a button if this thing they see matches the angle they are remembering. So it's a perceptual memory task, very simple. So we're decoding the relative angle between what you see and what you're remembering. So we run this model, and we have this plot is trials by time. So every color is a different state or decoder. And then you have this progression of states throughout the visual hierarchy. But as you see, there are differences between the trials. So look at this. So that's the average fractional occupancy, we call it, which is how much you are in each state per time point. And that, you can see the hierarchy, but there's a lot of variability. And then the question is that variability something that is meaningful, or is just noise? Because it could be, that's an estimation. So you have a variability that comes just because every time you estimate something, there is noise around it. So the critical result here is that we had reaction time. But before that, I'm showing here a histogram of the starting point of a given state and the finishing point, and you see there are quite a lot of variability, temporal variability around that. So the point now is that variability meaningful? Is that variability just noise-driven, or means something? So for that, we had reaction time. So we predicted using the speed at which you traverse through this sequence of states, the reaction time. And that was, there was a quite significant prediction. We also looked at the correlation between the state presence and reaction time, and you can see clearly this. So if you are in model 1, you can interpret this plot on the bottom. Like if you're in model 1 at 200 milliseconds, you have a strong correlation with reaction time. That means that this is going to be a slow trial. So your reaction time is going to be slow. That gives you a handle on something you wouldn't be able to observe otherwise. Because you have, of course, of course, you have the reaction time, but it's very difficult to know what's going on under the hood. So, and that's giving you a little bit of a handle on that. All right. I'm going to put a bit of math now. It's not free math. It's not it's not equations for the sake of equations. I'm going to make a point about this. So I'm going to describe the HM now mathematically a little bit. So it has 3 components in that equation. That equation is the likelihood. So we have X, which is the data, and Z, which is the state time courses, a latent variable in the machine learning jargon. And then the likelihood of that thing comprises 3 terms. One is the initial state for each segment of data. The second one is the transition. So we say the HM is Markovian because the state you are now depends on the state you were before. So in mathematical terms, if you knew the state you were before, then you are conditionally independent to all the other previous states. So this is Markovian thing. And that is parameterized by A, which is the transition probability matrix. So that's the first term is not very important, but that term is important. And then there's the third term, which is the probability of the data given the state you are. And that is where the choice of the state distribution matters. And that's how you can adapt to different modalities. So that is how we adapt to different modalities. And that's how we have many different types of HMM by tuning that model here. Just for examples, we can run a Gaussian distribution on power envelopes. So you are sensitive to correlation between power time series, for example. You can have, I'm not going to go into details on that, but you can have a Bayesian autoregressive model as a state. That means that you are very sensitive to spectral information. You can have what we call a time delay embedded Gaussian distribution, which is the one I presented before when I talked about continuous data. So that's also sensitive to spectral information, but you can include, it has fewer parameters, so you can model a lot of regions at the same time, whereas the autoregressive, you can only model a few regions, and you have the decoding model, for example. So the latest thing we've done, or we're working on now, will be soon released by Archive, is a generalization of all of these things. So we named Gaussian linear HMM. So the Gaussian linear HMM has this form. So Y is sometimes serious. We can think of, for example, pupillometry or some behavioral measurement that is continuous. And then that distributes Gaussian with the mean, this sum of two terms. So one is the mean parameter, and the other one is X by beta. So X could be the brain data, and beta is what relates X to Y. And then we have some covariance. So that is quite general, but with this model, based on the Gaussian distribution, we can model everything else that we've been talking about now. So the parameters of the model are again, the mean, the betas that relate X to Y, and the covariance. And every one of these can be modeled, non-modeled, so you cannot have it. It can be modeled as a per state, or can be modeled globally. So if you count, considering the covariance matrix can be diagonal or full, that amounts to 24 models. So 24 models comprises everything we've talked about so far and many others. So just an example, if that's the equation, the standard Gaussian linear HMM will have a mean parameter. That's, for example, power envelopes. That mean is how much power you have in that state. And then you have the covariance matrix, which is full, and encodes function connectivity. That's a standard Gaussian. And an autoregressive model, where you will have Y, the time series, model as a version of Y in the previous time points. That's an autoregressive model by definition. And then you have a diagonal covariance matrix. So that's an autoregressive model. Or you can have what I named the time-delayed embedded Gaussian, or HMM, which doesn't model the mean, but models the full matrix across a window, within a window. Never mind the details, but that is just a case of this Gaussian linear HMM. Or we can have a decoding model, where Y is the task data, X is the brain data, and then we have some noise, diagonal noise, which is basically encodes the variance of the error. Or we can swap it and have an encoding model, where Y is the brain data and X is the task data. So that would be like a GLM, basically, but in an HMM way. So the number of possibilities you have with this is quite a lot, actually. So you can model a lot of things with that, including relationships between physiological variables, for example, and activity in certain brain regions. Or you can model the dynamic relationship between a set of brain regions and another set of brain regions, which might be something that you are interested in. For example, you could have data set from the spinal cord and data set from the brain, data from the brain, and you have a pain experiment. You can get dynamics in the relationship between these two. So it's, again, 24 different models.
Are you presenting a toolbox?
I am presenting both. So this is, so as I said, this talk was meant to be sort of methods, like practical, not scientific. So yes, good point, thank you. So we had this HM for years as a MATLAB toolbox. So this Gaussian linear HMM paper and toolbox come together. So the toolbox is in Python, so we're trying to move to Python. So yes. And include, before I go on to that, given that we're talking about the toolbox, we put, well, I'm not going to talk about this next. Yeah. So, okay, so we're moving now. So we've been talking about brain behavior associations when we are talking about the task. So now we're going to talk about a different type of brain behavior associations, which is relationship with subject traits. Let's assume you have two cohorts. One is healthy controls and the other one has a clinical cohort. And you have a task or you have continuous data, a resting state or whatever, and you want to see how the dynamics of the characteristics of this brain activity differs between the two groups, for example. So in that toolbox, and we're going to talk about next, there is two ways of quantifying these brain behavior associations. One is statistical testing. There's a lot of routines for using permutation testing to test also task information, so whether your states relate to the task in that way or another way. There's a lot of routines about that, but I decided not to talk about this today. And instead, I'm going to talk about out-of-sample prediction. Out-of-sample meaning cross-validated predictions. So you have, for example, 200 subjects doing a task. You can, and there's two types of subjects, and you run the HMM on this task and see if there's a new subject coming, using those dynamics, you can predict whether that person is a control or a clinical, or belongs to a clinical population. Right. So there is a lot of interest in general with predicting individual traits, not just clinical, but also psychological, like intelligence and things like that. A lot of that is being done in fMRI. But I think there's also interest in electrophysiology. So basically, we have brain data across subjects, and then you have something you want to predict. And that is a difficult problem in itself because it requires a lot of feature engineering. So you cannot use the raw data as features for prediction because that doesn't mean anything. So you have to create features out of this. And that typically is done sort of manually. So I'm going to talk about how to use the HMM to create features on those data sets that are useful. So that's not new. So every time you predict things from brain data, you will always use an intermediate model of the brain data for prediction, just because the raw data you cannot use it directly. And those models range in complexity from some simple models to very complex models. Like, for example, a very simple model, and the one that is used the most in fMRI, for example, is functional connectivity. It's just a correlation matrix. There's a very, very simple, you can call it model if you want, but it's just a very simple description of the data. So you compute the correlation per subject, and you use those correlations as predictors. That's the simplest. Or you can have very, very extremely complex models where you have a lot of biophysical assumptions of how the brain works, and you put those assumptions in the model, and then you try to use those for prediction. In between, you have, and that's what I'm advocating, in between you could have models that are less biophysical, simpler, but still encode some information about dynamics and the way the brain moves from one brain place to another. So the HMM is one example of that. So we run the HMM on a group of subjects, and then we have these parameters. So now the question is, and what I'm going to talk about now, is how can we predict from those parameters some subject trait like intelligence, for example. So I'm going to tell you briefly about kernel methods before I move on how we do this from the HMM. That's if you have a degree in computer science or you have a bit of a mathematical inclination, you know kernel methods, but I'm going to explain this quickly for everyone. So let's imagine that you have a matrix like number of subjects, for example, a number of features, and it's X, and you're going to predict some variable Y. The standard way of doing it will be a linear prediction. So you use every feature of Y gets a beta coefficient. But sometimes a linear prediction is not sufficient, so you want to have something more non-linear that accommodates the data better. And the most simple way of doing that will be to use expanded terms on this column of x. So you could use, for example, a polynomial expansion. So you have x to the square, or maybe interaction terms between columns. So you expand your space into something that has more variables, and then you perform a linear prediction on that expanded space. And that gives you, using linear machinery, that gives you a nonlinear prediction. So the kernel methods for prediction is a way to do this more conveniently. So the trick, what is called, actually is called the kernel trick, is that there are kernel functions. A kernel function is something that takes a subject and another subject and gives you the similarity between these two. That's a kernel function. With a kernel function, you don't need to do this expansion explicitly. So there is a correspondence between this expansion of x and a kernel function in a way that you don't have to do this explicitly. And that is computationally advantageous. And sometimes you can't, depending on how you want to do the prediction, you can't even do an expansion easily. So point being, if you have a kernel function that tells you how this subject and this subject are similar, you can construct what is called a kernel matrix, subject by subjects, and use that for prediction. So you move from your original data, which is subjects by features, to a kernel matrix with subjects by subjects. That's the essence of kernel learning. So where does this bring us? A trivial way to predict from HMM parameters. So we could sort of take every HMM parameter and use it as a feature, and you can construct an X matrix, subjects by number of HMM parameters, and just predict on that. That ignores the mathematical structure of the HMM. And that has consequences that I will show you in a second. So as a way to create a method from prediction that takes into account this mathematical structure, and we'll see how is that beneficial, we devised something called the Fisher kernel. So what is the Fisher kernel? The Fisher kernel is a kernel function that tells you how two HMM models are similar, to which extent they are similar, so that we can construct this kernel matrix subjects by subjects and then perform prediction on that. So a bit more in detail, the way this works is, so you have your data, EEG, fMRI, or whatever you like, across subjects. You run HMM at the group level, so you can one HMM for the whole cohort. That's what we normally do. Then there is a process that we call dual estimation, where you adapt this group level HMM to each subject. So every subject now has its own HMM set of parameters. So that's this. And then we apply this Fisher kernel thing on these parameters to create this kernel matrix. And then we perform prediction using anything that you like. So you can use support vector machines. We use kernel-rich regression, but it doesn't matter. Should I say what is the Fisher kernel? All right. So just very quickly, so you're wondering what the Fisher kernel is. The Fisher kernel is, so you have your HMM individual estimations, and you have your group HMM. So the Fisher kernel is the gradient between your individual HMMs and the group HMM. So that is how much you have to push every parameter from the individual HMM to meet the group level HMM. So that's gradient space, and that preserves the structure of the model. It's only interesting if you're interested in the mathematics of this kind of thing. When it comes to practical aspects of this, we're going to evaluate this physical. Yes. Yes. Your question is how to get the individual HMMs. So we have, remember the HMM has state time courses. So those are individual, subject specific. And then you have the state descriptions that are grouped at the group level. So what you do is you get the state time courses of that person, and then you re-estimate the state parameters on the data of that person only. That gives you a state subject specific state parameters. So you already had the time in the causes. Now you have the state distribution as well. So that is very simple. It's like if you work with ICA, it's like dual regression. It's exactly the same. Yeah. Right. Thank you. Thanks for the question. Right. So we're gonna see. You might think, oh, what's all of this mathematics? What is the point of that? You're gonna see that actually makes a difference. Like doing this properly, and by properly I mean respecting the structure of this mathematical object that is the HMM, how that pays off. And it doesn't matter for you, it's just a function to use. We're gonna think, like when people do a prediction, or report prediction of individual traits, they normally report accuracy. One of the points we're making in this paper is not just to look at accuracy, is to look also at reliability. And by that, we mean not having extreme errors in the prediction and robustness. As an example, why those things matter, I don't know if you've worked on this, but there is a lot of interest in predicting brain age. So the way the field goes about brain age, which is a big thing now, is that, so you have a cohort of people, everyone has the biological age, then you predict their age. And like, for example, if you are 50, and your prediction is 60, that's an error of 10 plus 10. So that is not just a statistical error. It's not just that the model was not exact in predicting because of the estimation error. It also means that your brain age is likely to be higher than your biological age. On the other hand, if you are 40 and you're predicted a brain age of 20, that might be an indication that your brain age is actually younger than your biological age. And people that have worked with clinical populations have verified this in practice. So when you have an ALS population, for example, people have worked with brain age prediction in ALS. The predicted brain age for ALS is always the error is systematically positive. So the predicted brain age is always higher than the biological age. So the brains are older. So there is a lot of interpretation in these errors. But you can only do this interpretation if the errors are not crazy. Sometimes when you work with age prediction and you have, you're experimenting with your methods, you get extreme errors. You get someone who is predicted 500 years old. That is not, you cannot interpret that with that person has an old brain. There's something intrinsically wrong with that prediction. So you want to avoid that. So we looked at the maximum absolute error, and we want to have that under control. So that's what we refer as reliability. The third point is robustness, which is related, but it's not the same. So if you're a clinician and you are going to do this brain age business as a biomarker, you want the prediction to be robust. You want that if you change the data a little bit, or you change your pipeline a little bit, you don't want the prediction to change a lot. That's obvious, right? You want things that are, perform consistent. So for that, we're doing cross-validation. So we altered the structure of the faults. So we changed the training data and see how the errors change as you change the training data. And we want this not to change a lot. So that's robustness. So we evaluated different kernels and methods. Some of them include the structure of the HMM as a mathematical, respects the mathematical structure of the HMM, which is the Fisher kernel, and some don't. For example, these naive kernels, they just vectorize the HMM parameters and do some naive or vanilla prediction on that. That does not respect the structure, the mathematical structure. So first of all, you see the Fisher kernel is more accurate. So it just gives better prediction on average. So that's what everyone reports. So that's nice. This, by the way, this is predictions across a range of cognitive variables that are typically difficult to predict. So don't be too put off by the fact that we are predicting 20% explained variance. That is kind of the standard in the field. It's difficult to predict intelligence. Then when you look at reliability, so we can look at quantiles. So you have theoretical quantiles in the x-axis and the maximum error on the y-axis. So you want that thing to not go super crazy at the right of the plot. So you can see that when you do not respect the mathematical structure of the HMM, which is the naive kernels, the red and orange, at the right of the plot, the errors are crazy. So this is predicting age, and it's an error, the maximum error of 1,000 years. Maximum error of 1,000 years. So it performs okay most of the time, but sometimes it just goes nuts. So you don't want that. And the physics kernel, in fact, the worst errors, they are never very big. So on average, might be a little bit better than the naive kernels, but the important thing here is that it doesn't go crazy in the most extreme case. So that's important. And the third one is robustness. So we repeat this cross-validation using random faults a number of times. So these boxes reflect the variability across faults. So the size of those boxes is what we're looking at now. So the green is the physics kernel, and you see the box is small. So that means that if you change the structure of the faults, the prediction is kind of consistent. That again, I think is reflected, is due to the fact that you are respecting, you are mindful of this mathematical structure, and that pays off, giving you a robust estimation. All right, I'm almost done. So yes, Frick, one of the points of this talk was to present the HMM toolbox, and that's my last slide. So it's not, I mean, it's on GitHub, there's no paper out there yet, but we are hoping to put this up in the next 1 month or so. So the take-home messages of this is that it's versatile. So you can implement almost any model you want as far as it's based on the Gaussian distribution. So that's 24 models. You can use different types of data, brain data combined with data that is not brain data, or even non-brain data at all. So you can have almost anything you want. You can have your behavioral experiments with pupillometry and cardiac rates if you want. We have put a lot of effort in implementing routines that make this inference of the HMM efficient. I didn't talk about that, but it uses something called stochastic learning. So that basically, it doesn't matter how it's done, but what it matters is that it can handle very big data sets at low computational cost. So we run this in the UK Biobank, for example, 500,000 subjects, 100,000 subjects, or the HCP, which is also big, and it runs in 1 hour or 2 hours without using a lot of memory. And then particularly we put an emphasis on utilities for doing permutation testing and out-of-sample predictions. So basically to relate these HMM parameters to behavior and think in ways that are easy and interpretable. And with that, I thank you all for coming. Sorry? Oh, yes. You just want to do it? No, I don't know how to do it. So you mean online?
No.
Oh, okay. I thought you want to look at, yeah, of course. Yes.
So thank you, Diego, for your talk. And yeah, please let us know if you have any questions.
So for the model fitting procedures, I didn't quite catch this because you did talk about some Bayesian models, although I don't know if that meant Bayesian in the sense of model fitting or Bayesian in the sense of decision making or something. But so then is your toolbox, one question is the toolbox Bayesian, because I didn't quite catch that, or is it based on classical statistics? And two, did you do Bayesian methods in effect?
Okay.
Questions so people online can follow.
Right. So you asked if what did I mean by Bayesian and whether the toolbox is fully Bayesian, basically. Because when people talk about Bayesian, there's neuroscience connotations on that. And I did not mean any of this. What I meant is that so when you have a model, the model can be Bayesian. That means that you have priors. It's a probability distribution with priors, and so the model is Bayesian, and it uses Bayesian inference. Bayesian inference means just that you have a Bayesian model and you find the parameters in a way that is compatible with that. So we have routines for estimating these parameters that are Bayesian. But yes, I did not mean anything neuroscientific in terms of Fristonian things. The HMM is a Bayesian model. Yes.
The previous correlation slide.
I'm showing posterior distributions. Yes, yes, but you have the full distribution, the full estimation. Yes.
Models have a lot of transition probability across all states. Is this a problem for the estimation of the model, that this fact that you can basically move from one state to any other?
So I didn't talk about that, but you can restrict that. So you can, in the toolbox, you can, for example, if you have an hypothesis visual system that I go throughout a sequence of states, and that's your hypothesis, you can impose that structure in the model. So you can impose any structure on the model and forbid any transition that you don't think you want to model. That's one thing. And another thing that is relevant about transitions is that you can also, there's a parameter which is and that relates to the things that you do personally. There is a parameter that you can use to encourage more or less transition in between states. So it's the prior distribution of moving from one state to another. So that's sort of a way of, so you work with the same hidden Markov model. The problem with the HMM is that you have a lot of spurious transitions. So every time you have a time point, you estimate whether you move to another state or not. Then there is sometimes you move to another state when you should have not moved to another state. So the one way of going about that is the semi-hidden Markov model that will be talked about in the next session, I think. And the other one, simpler one that we use, is just to have this prior probability of transitioning a bit lower. But yeah, both things can control the dynamics of the states. So, okay, so how does your group, how do you compute the group level estimation? How that relates to your subject level from which you get these gradients? Is that? Yes. So the group level is the first step. So you have data from 100 people, you run the HMM on everyone. So you have state time courses for each person, and then you have a group level estimation of the state probability, the state distributions. So from there, you go to individual level estimation. So you have the state time courses were individual in the first place, and with those you recompute the state probability, the parameters of the states for every person. So the Fisher kernel, I think that's your question. The Fisher kernel is the gradient between the group level and every individual. So you got a gradient per person, per parameter, and that's the distance between the group level and the subject level for each one of you. Is that answering to your question? Okay. He's asking how it's unclear to him how you get the group estimation in the first place. That's very straightforward. So you have time series for every person. You just concatenate all of them, and you run the HMM. So of course you take into account when one session finishes and the next starts, but you just run the HMM on all the data together. That's a group level estimation. Yeah.
You're going to get participants that have one state, and another participant that is a different state, or that doesn't happen.
That's so there's another problem, which is you can have a good estimation or a bad estimation. If your for example, if your pre-processing is not very good, if you didn't register your subjects very well, or anything like this, you can have states, subjects being described by a single state. That is what I will name a bad estimation. We don't want that. There are ways to go about this. We have a paper actually on this. It's a technical problem. If that's your case, then something's wrong. He's asking about, because you have a group level estimation and subject level estimation, he's asking about whether that is Bayesian hierarchy. So you have a sort of a group level, and then, that is a proper hierarchical modeling of the group and the individual. We've done work on that, not on HMM, but on ICA. So you look at probabilistic functional modes, PFMs. We use that idea. So we have a group level, and then you estimate individual level, individual specific versions of the ICA maps. And that is all together in the inference. That's a proper Bayesian hierarchical thing. This is not like that. This is complicated, by the way. It's not trivial to do, and it makes the estimation quite hard. So we do not do that. So we have estimation of the HM at the group level. When it's done, it's done. And the next step is this dual estimation, which is very simple. So what you're saying is complicated. What I'm saying is very simple, but you can do it. But it's difficult.